Hi, ladies and gentlemen. If you guys need some help with the strawberry DNA extraction lab, hey, I just wanted to go through a couple things with you guys. So, first of all, if we read through the background information here at the beginning of the lab, you guys will see it says DNA is found in all living cells. No other substance is as important as DNA. It contains a set of chemical instructions that controls all the cell's activities. It also carries coded information in its nucleotide sequence that makes every person unique. DNA is a double stranded macro, that means large molecule, composed of long chains of nucleotides. A nucleotide is a phosphate group, a deoxyribose sugar, and a nitrogenous or nitrogen base. The nucleotides are linked together, forming a long, twisted ladder with a backbone consisting of alternating sugar and phosphate units. Hey, and that twisted ladder is also called a double helix. Hey, the rungs of the ladder are made of pairs of nitrogenous or nitrogen bases that are held together with what are called hydrogen bonds. In DNA, adenine pairs or bonds with thymine, guanine pairs or bonds with cytosine. The sequence or order of the nucleotide bases provides the instructions for how to build proteins, which allow our bodies to function and have particular characteristics. Prior to cell division, cells replicate or copy their DNA. DNA replication involves the two strands of the double helix, or the two sides of the DNA ladder, separating, and each strand serving as a template from which to make a complementary strand of DNA. Okay, so what that means is the double helix, hey, you've got your DNA that's twisted up. Well, it consists of two strands that are linked together. Well, when DNA replication occurs, the DNA is going to open up, and then new bases will come and pair with the bases on this side, and new bases will come and pair, with the bases on that side. And then we'll have double stranded DNA over here, double stranded DNA over here. I have an exact copy of the original double stranded DNA. Okay. So that's called DNA replication. Okay. So DNA replication. And when we talk about complementary strand, what I mean by complementary is that on the template, there's an adenine. Okay. The new base that will pair up with it that's complementary is thymine. Okay. If the next base is guanine, the new base that's complementary to that is cytosine. Okay, and lots of times we just abbreviate instead of saying the entire base name. Okay, we use A for adenine, T for thymine, G for guanine, C for cytosine. Okay, so DNA can be extracted from cells using a simple technique using household chemicals. Okay, those household chemicals, are things like our detergent, and salt and water. Okay, the process of DNA extraction is an essential first step to the analysis and manipulating. Okay, remember we talked about in class about how we can use DNA then to make new an organism that has a new trait in it. Okay, or we could potentially fix an error in the DNA. Hey, scientists extract DNA to detect gene defects or diseases, produce DNA fingerprints, or make recombinant DNA organisms, or organisms that have DNA from more than one source. The process of extraction consists of the following steps. The cell membrane is broken down using the detergent and salt. Okay, the detergent disrupts the lipids or the fats that make up the cell and nuclear membrane, causing them to open up. Okay, and then the salt helps to neutralize the charge on the DNA and remove the proteins that are attached to the DNA. Okay, this makes the DNA less likely to dissolve in the water and makes it easier for it to precipitate or solidify, the DNA out of the solution. Okay, and then when we add the isopropyl alcohol, that's the fancy name for rubbing alcohol, to we do that to precipitate or cause the DNA to leave the strawberry solution and go into the alcohol solution. So we've got, these were essential questions, things that we were thinking about while we were doing the experiment. Hey, so if you go all the way down here to the bottom, these are the questions related to the lab. Hey, so where can DNA be found in eukaryotic cells? Remember, a eukaryotic cell is a cell that is a little more complex. So it has organelles, including the mitochondria and the chloroplast and the nucleus. So when we talk about our plant cells and our animal cells, they have a nucleus. That's the control center of the cell, and that's where most of the DNA can be found in our eukaryotic cells. Our second question asks about what's the purpose of the detergent? Okay, what does the detergent or the soap do? Remember, the soap is like the little scissor. Okay, and that soap is going to take the cell membrane that's made of lipids and chop it up into little pieces and allow us to open up the cell and open up the nuclear membrane. Question number 3, what was the purpose of the sodium chloride? Sodium chloride is the fancy name for salt. Okay, so sodium chloride, its job was to neutralize the DNA. It does that by attaching to phosphates on the DNA, and then that allows us to precipitate the DNA out of the strawberry juice. Okay, why was the isopropyl alcohol, why was the rubbing alcohol added to the soap and salt mixture? Okay, we added it to pull or draw out the DNA, to get the DNA to leave the strawberry juice and separate from the other parts of the cell, hey, that are kind of left behind in that soap salt mixture. Hey, describe the appearance of your final product. Hey, its color, physical characteristics, right? So what does it look like? And what did it remind you of? Hey, you guys can write up a description of that. Hey, and then now we get to questions about DNA and its structure. Okay, you should be able to answer these using the background info or even the science notebook info. So what is DNA made up of? DNA is a long chain, and it's a long chain of nucleotides, long chain of nucleotides. Then it asks how many strands make up DNA. So is it single stranded or double stranded or triple or quadruple? Hey, well, DNA has 2 strands, remember, and they're held together by hydrogen bonds. What are 5 characteristics of DNA? Okay, 5 characteristics. There are probably lots of characteristics, but I would go back to this first paragraph. Hey, what are some characteristics of DNA that are described here? Hey, well, it's found in all living things. Okay, 2, it codes for protein. 3, it's double-stranded. 4, it's made of nucleotides. Nucleotides are phosphates, sugars, and bases. Hey, its shape, it's a double helix. Hey, you could also say that it has adenine and thymine paired together or bonded together. Guanine and cytosine bond together. Okay, so those would all be examples of characteristics of DNA. Number 9, what holds one strand of DNA to the other in the double helix? So what's holding together the adenine and the thymine? What's holding together the guanine and the cytosine? Hey, if we read up here, hey, the rungs of the ladder are made of pairs of nitrogenous or nitrogen bases. They're held together with hydrogen bonds. Hey, then the next one, question number 10, when do cells duplicate or replicate their DNA? Again, we're going back up to that first paragraph. Okay, and we're looking for DNA replication. Hey, that's in these last 2 sentences. Okay, those last 2 sentences tell us when DNA is replicated. Okay, so you read that section there, and you should be able to answer when the replication occurs, and then how do they replicate? Okay, how do they replicate is described really in the last sentence here. Okay, and there is a description also, like I said, in the science notebook reading, so you could use that to be even more detailed. Okay, but definitely this last section last sentence of the first paragraph, okay, will help you answer how they replicate. So it says DNA replication involves the two strands of the double helix separating, each strand serving as a template from which a new complementary, and remember, complementary means that the adenine would pair with the thymine. Okay, guanine would pair with the cytosine. Thymine would pair with adenine. Okay, so that's the DNA extraction lab. Conclusion questions. Once you have all those questions answered, make sure you've answered them in complete sentences, CSIQ. Okay, and then please turn that in and hit submit and have a fantastic weekend.